Hello, it's my pleasure to speak with you today during the EPI webinar series on sudden unexpected death in epilepsy. During this presentation, I'll be touching upon something I find to be very important in advancing our knowledge of the mechanisms, prevalence, risks, and hopefully mitigating the causes for sudden unexpected death in epilepsy, SUDEP. And this requires a multi-system approach that doesn't only include the brain, but includes other important areas of the body, such as the heart, the lungs, and various other autonomic nervous system of the body, as well as the importance of taking a multidisciplinary approach that includes experts in many areas of medicine, as well as families and advocates to really raise awareness about SUDEP and to advance our understanding of SUDEP. In epilepsy, we oftentimes focus on the various psycho and social comorbidities related to epilepsy. But what I find the most scary about epilepsy, and that it's oftentimes very underreported, is that people with epilepsy are at a 24-fold increased risk of sudden death. And to make this even more scary, oftentimes the cause of this sudden death is unknown and is termed SUDEP, sudden unexpected, non-traumatic, non-drowning death in an individual with epilepsy. It may be witnessed or unwitnessed, in which the post-mortem examination does not reveal an anatomical or toxicological cause for a sudden death. And in SUDEP, it accounts for 7.5 to 17% of all deaths in people with epilepsy. So in a general population with epilepsy, about 1 in 1,000 people with epilepsy will die of SUDEP each year. And this number is even higher in some of the intractable forms of epilepsy, the hard to treat forms of epilepsy, up to 6.7 per 1,000 people per year. And even in some of the most severe genetic forms of epilepsy, it gets all the way up to 14. Due to these high risks of sudden death, we've focused a lot on what are the risk factors to identify the at-risk populations. And as you can see here, there's numerous what are considered non-modifiable risk factors, such as unclear treatment history, a high rate of convulsive seizures, particularly greater than 3 generalized tonic-clonic seizures per year, an increased seizure frequency, and long duration of epilepsy. But there's also many modifiable risk factors, such as your sleep position. In a prone position, puts you at a higher risk of SUDEP. Also, nocturnal monitoring. Those who have monitoring during the night, there's a decreased risk of SUDEP. Also, being compliant with your anti-seizure medications and limiting alcohol use. So a couple years ago, there was a study that compared those without versus with a history of generalized tonic-clonic seizures. And as I mentioned above here, those with a history of generalized tonic-clonic seizures, there was an 18.65-fold increased risk of SUDEP. If there was versus no nocturnal monitoring, you can see there's a 3-fold increased risk in those who did not have nocturnal monitoring. But the biggest concern here was that there was a 67-fold increased risk of SUDEP in those with a high risk of generalized tonic-clonic seizures, your convulsive ones, and no nocturnal monitoring. And I highlight this because for a while, there was a lot of debate whether to counsel about the risk of SUDEP. And I take this quote from his 2020 paper where it says that it was not that long ago that some believed there was a little point to discussing SUDEP in patients. Why cause such anxiety, it was argued, if the risk could not be influenced? And as I've shown you on this slide here, that was completely incorrect. There are lifestyle decisions that influence your risk of SUDEP. Having the nocturnal monitoring, sleeping in a non-prone position, being compliant with your anti-seizure medications, which we know will reduce some of these considered non-modifiable risk factors. If you're compliant with your medications and they're working, it should, hopefully will reduce the frequency of your seizures here. So we've talked so far about some of the modifiable and non-modifiable risk factors, but none of these truly explain why people are dying of SUDEP. So I'm going to transition now to talk about some of the proposed mechanisms for SUDEP. The first one, obviously starting with the brain here, is cerebral electrical shutdown, or hypoperfusion of the brain, meaning you're not getting enough blood flow to the brain during the seizure or after a seizure. The next one are respiratory disturbances. A lot of researchers have demonstrated that there is respiratory dysfunction, particularly right after the seizure. The failure to take that gasp after the seizure, the lack of restarting your breathing after the seizure. Also, as I mentioned on the previous slide, sleep position or sleep disturbances. For example, if you're sleeping in the prone position with your face into the pillow there, if you're not having a seizure, hopefully you will wake up, whether you remember or not, to some level of consciousness to lift your head and then to turn it. But obviously, after a seizure, you're in this post-ictal state, or unfortunately, even during the seizure, you're not able to have that level of consciousness to lift your head and to turn it. So there's in essence, that suffocation or asphyxiation in the pillow there can put you at an increased risk of SUDEP. And that could explain one of the mechanisms. The fourth one is the autonomic dysfunction. So the autonomic nervous system is the neural connection between the brain and all areas of your body. The autonomic nervous system's, for example, that fight or flight, that sympathetic, branch. If you suddenly get startled, there's many things in your body that have to react to say, we got to increase our heart rate because we got to run. Something just happened. Or, during sleep at night when we're resting there, our heart rate drops down. Also, the autonomic nervous system innervates your gastrointestinal system that helps with digestion. So dysfunction of this autonomic nervous system, particularly how a seizure could emanate down to dysfunction in the autonomic nervous system that can lead to further downstream problems, is your fourth proposed mechanism for SUDEP. And the final one, which is a major focus of my lab, is electrical disturbances in the heart, your cardiac arrhythmias. So as I showed you, there's many proposed causes for SUDEP. And what I'm going to really emphasize now is there is no one single cause of SUDEP. We do not understand the whole cascade that's developing in cases of SUDEP. And in 2013, Philippe Rivlin and numerous others demonstrated that SUDEP is a multi-system cardiopulmonary shutdown of the body. And you can see here are numerous unfortunate cases of SUDEP that were captured when people were in the epilepsy monitoring unit. And the top line for each patient is changes in respiratory function, and the bottom one is abnormalities in cardiac function. And you can see there's a lot of overlap in each, which demonstrates that this is a multi-system cascade of apnea, stopping breathing, heart rate decreasing in terms of bradycardia, asystole, no beats, the heart stopping, and then unfortunately the terminal asystole and terminal apnea as well. So I've spoken so far about some of the risk factors for SUDEP, as well as some of the proposed mechanisms. And then for the rest of the talk, I'd like to focus on some of the ongoing research and where the field is going right now to help understand and prevent SUDEP. So the first one, as I've been alluding to throughout, is the importance of taking a multi-system approach to understand this cascade of neuronal, cardiac, respiratory, autonomic, and likely many other pathologies that are culminating in SUDEP. The next one is the multi-scale approach. And on the next several slides, I'm going to be giving some examples of this. But there's research into SUDEP that's at the molecular level, trying to identify certain genetic risk factors. For example, the group in Australia collected DNA samples from many SUDEP cases and identified numerous genetic variants that are associated with an increased risk of SUDEP. The next one, going from the molecular up to the cellular level, single cell. A lot of researchers are demonstrating that abnormalities in the normal functioning of the cell, particularly the electrical function of the cell, indicate potential mechanisms for SUDEP, where you have alterations in ion channel function that leads to these electrical disturbances at a single cell, which then could result in thus abnormalities at the whole animal or whole patient level. And this has been modeled, for example, in mice and zebrafish, which provide a very high throughput means for evaluating, various genetic variants, as well as also the effect of various medications. And then there's also work that's being done in larger mammals that better model the human physiology. Going up to the next level, we have our clinical databases. For example, there was a Center for SUDEP Research database, which included long-term EEG and ECG recordings from people who come in the epilepsy monitoring unit. And this provides a wealth of information for doing deep dives and data queries into the whole cascade and what is unique to some of the people who unfortunately later die of SUDEP. These long-term recordings can help us identify some of these risk factors and at-risk populations. Also computational. We've been hearing a lot about AI, your artificial intelligence, machine learning models, which sound very abstract, but all this large amounts of data enable us to feed them into these models to then hopefully make predictions of who may be your at-risk populations and what are the unfortunate combinations of various substrates and triggers that ultimately may result in SUDEP. And then all this is important to understand the mechanisms and risk factors, but we need to then use the information to then develop devices that will alert us when an emergency arises, or ideally even prevent these potential events. And there's a lot of development recently in terms of wearable monitors, stimulators, and changing a person's position to put them into a safer position, after a seizure, that non-prone position. So I'm going to provide some examples of this here. In my laboratory, we really focus on looking outside the classic organ of study. So we know that various genetic ion channel mutations or variants can lead to electrical disturbances, in the brain in the form of seizures, or electrical disturbances in the heart in the form of arrhythmias. But they're not mutually exclusive. That a ion channel mutation could lead to alterations in electrical function in both the brain and the heart. So starting at the patient level here, I asked several years ago, do people with this classically studied cardiac disease, long QT syndrome, are they at an increased risk of not only arrhythmias, but seizures? And using a large clinical database, we were able to demonstrate that yes, people who had long QT syndrome were genotype positive were at an increased prevalence and risk of having a history of seizures. And we dug even deeper into the various types of long QT syndrome. In the green bar here, the long QT2 group, we showed were at the highest risk of developing a seizure and having a history of epilepsy. And we even dug even deeper into where these genetic variants might be in the ion channel. And further, we went on to show that people who had a history of not only long QT syndrome, but a history of seizures had worse cardiac abnormalities. So all this here are associations, and they really provide a very strong clinical significance for future work. And that's where we're going right now, to take these results at the patient level and now bring them back to the lab and ask, why are these people with long QT syndrome at an increased risk of seizures? Why are these people with long QT syndrome who have a history of seizures having more arrhythmias? And this is an example of this multidisciplinary type approach, where we're taking results from the patient level and bringing it back to the bench. Also multi-system, where we're taking a classically studied cardiac disease and looking outside the heart, looking at the brain here. So we generated the first knock-in rabbit model of long QT2. And this, to explain this a little further, we went into the genetic code of this rabbit and modified it to model what's seen in people. And using that, we can take this multi-system approach that we're doing work at the molecular level, studying, is this mutant channel present? What effect does it have upon the single cell, properties in both neurons and cardiac myocytes? And then going up to the whole animal level, where we record continuous EEGs and ECGs to hopefully understand this cascade. So I emphasize this multi-system type approach. So this is exactly what we're doing in the lab here, where we will record from numerous rabbits at the same time, and we place electrodes. Just like when you go to the doctor, your standard EKG electrodes, as well as some electrodes on the scalp, your EEG electrodes. And we record simultaneous ECGs, EEGs, and then they have just like a little nasal cannula there that's monitoring their breathing activity as well as their oxygen saturation. So we can really understand the multi-system cascade of events that arise surrounding a seizure and leading up to sudden death. And here are some recordings where you can see the EEGs, the cardiac ECGs, the oxygen saturation, and your respiratory patterns as well. Now, we couldn't have a model of long QT syndrome without demonstrating that we actually have the QT prolongation on the ECG. And in fact, we did show that our mutant rabbits have prolongation of the electrical activation to recovery process in the heart there. And that's just an example to show that using these recordings from the rabbits here, we can dig deep into doing more advanced analyses to really quantify these changes. As we're getting EEG recordings, we also are able to look for the presence of epileptiform activity. So for example, in one of our rabbits that do not have the mutation, we see very normal EEG patterns, but in some of our mutant rabbits, we see these epileptiform spikes here. And you can see how this one grows and grows until it's hitting many areas of the brain. Here is some other examples of EEG discharges, and here is an actual seizure that we caught in the rabbit here. And as shown right here, we can also see that our mutant rabbits, we're able to quantify that there's a higher prevalence of our mutant rabbits developing epileptiform discharges and seizures compared to the wild type ones, where we do not see any seizures. Because of this continuous monitoring, we're also able to capture the multi-system cascade of events that leads up to sudden death, where we can see, in the EEG, the rabbit develops some myoclonic jerks, some short periods of generalized tonic-clonic seizures, and then we led up to this very large seizure here, and you can see we recorded up to the seizure, and then after the seizure here, we see a huge change in the EEG and ECG patterns during this post-ictal period, where the heart rate dropped a lot, and there was EEG suppression, and ultimately this rabbit did not recover from the seizure. And while this was very unfortunate, it provides a lot of insights into the unfortunate lethal cascade of both neuronal and cardiac abnormalities that led to SUDEP. So to put all this together, here are several cases of sudden death that developed in our rabbits here. And for all of them, we had this convulsive seizure, this dark green generalized tonic-clonic seizure. And then after the seizure, the light green indicates this postictal generalized EEG suppression. But then the purple, the respiratory, and the blue, the cardiac, we can see that there's a whole cascade of events that are happening concurrently, and some at the same time, some one after the other, leading up to where the star is, eventually the heart stopping, and eventually death. So while I've talked about many cardiac and EEG measures and abnormalities, the real take-home from this here is that we have to take a multi-system approach, that we need to look outside the brain, and we need to get having simultaneous recordings from the brain, the heart, the lungs, all these areas to really understand this cascade. And while these events are very unfortunate, it helps us to better understand what's happening so we can develop preventative measures to prevent future cases of SUDEP. So I'd like to give you another example of taking a multi-system and multi-scale approach to researching SUDEP. So the previous example I gave you, we started at the bedside, at the patient level, and translated those results back to the bench to understand the mechanisms and the cascade that surrounds SUDEP. Now I'd like to, for the second example, to start at the bench level. So using a severe genetic form of epilepsy, we worked with animal and cellular models of this disease and looked at in these cells, they're from the heart, not only in the brain, but in the heart, they were hyperexcitable. And this leads to cardiac arrhythmias. And we actually captured sudden cardiac death in these mice here. But a critical eye might say, well, these are little mice. Do they really model what's happening in people? Or these are cells in a dish. How well does that model, the patients? So using large clinical databases, like I mentioned earlier, like the Center for SUDEP Research Database, where we have long-term EEG and ECG recordings from several thousand patients, as well as using wearable technology where we get continuous cardiac ECG, respiratory, and even oxygen saturation recordings, provides a wealth of information. So we feed this into our computer programs, into our algorithms here, and using these algorithms, we hope to then develop pathological markers for SUDEP that we can then ultimately take full circle and validate back in animal and cellular models. So I'd like to dig a little deeper into how we analyze some of this data here, because I find this to be very important for really evaluating the risk. So oftentimes we take measurements during a static time point. For example, I went to the doctor at 3 p.m. today and I got this recording, and hopefully they gave me a clean bill of health. But there's unfortunate circumstances where the person leaves the doctor, everything looks good, they're running to their next appointment, they're nervous, they're rushed, and then something happens out in the parking lot. And you say, wait a second, that person just got a clean bill of health. The recording at 3 o'clock today looked great. Well, what happened 15 minutes after that? What happened several hours later? Maybe while they were sleeping. So we take a much more comprehensive type approach where we take recordings throughout the 24 hours of the day. Like here would be midnight, here would be noon, and you can see how this fluctuation in this cardiac measure throughout the day. So the red circle here, if you're inside that red circle here, that's normal. But you can see at several points of the day, this patient's recordings indicated that they were going into a prolonged QT, meaning an increased risk of a substrate for arrhythmias here. So it's really critical to look at this under many different physiological states. For example, when a person is sleeping, during times of minimum, middle, or median, and maximum heart rate, as well as leading up to the seizure and then after the seizure. There's a huge amount of changes that arise surrounding these seizures. So we need to take the recordings throughout this entire time period. So to get these long-term recordings, we need technology that will record this throughout the entire day. And thanks to a lot of advances in technology recently, we've had many new wearables that have come out on the market, such as watches, vests with detectors in them, as well as patches that you can stick onto your skin. Also, there's numerous research and clinical grade implants that provide continuous monitoring. So what are these things measuring here? Some of the biosignals include accelerometry, such as your movement. We think of the accelerometers in our watches detect when we're exercising, but some of these accelerometers also can detect when there's large motor movements, such as around a seizure, electromyograms, which is changes in skin conductance here, and electrodermal activity. Both of these together give measures of autonomics and sympathetic activity. Next, heart rate and heart rate variability. Heart rate variability, again, gives an indirect measure from the heart, but of autonomic function. And also keeping logs of circadian cycling and sleep recordings. All this can provide insights into some of the conditions that may all come together to set up the substrates for seizures and, unfortunately, even sudden death. So we need to take those recordings and information from them and to develop alarm systems or even ultimately interventions. So for example, an alarm may trigger a family member to provide care, such as CPR, or to stimulate the person, or to change their body position. The final arm for this multidisciplinary approach is the critical need for a partnership between the families and researchers. So to me, the parents' passion and dire request for new information stimulates us to want to advance our understanding of the causes for SUDEP and drive us to develop new detection tools and ultimately identify new therapeutics to prevent SUDEP. So to me, whenever I speak to the families, I find their dire request to be very contagious and make me want to, get back in the lab and it really puts all of the work that we're doing into perspective. And I encourage all the people from my lab to come to some of the EPI events because it helps us to really see what we're working for each day. So to wrap up today's talk, I just want to summarize a couple key points. That SUDEP is a devastating event that is a result of a complex cascade of multi-system changes. And to advance our understanding of the mechanisms and risk factors for SUDEP, we need to instill approaches that span from the molecular to the cellular to the animal models and all the way up to the clinical level. And we need to develop, models that will reproduce the human neuro, cardio, respiratory pathologies, which ultimately set the stage for these future therapeutic testing. Also, comprehensive assessments of biomeasures during various physiological states and surrounding seizures will help us to better understand these events. And to conclude, to minimize the prevalence of SUDEP, we need to develop and implement using various wearables and seizure alarms to alert caregivers and to maybe even develop technology that can even intervene to prevent these lethal events. And all this can only be accomplished with the continued partnership between the families and researchers. So thank you very much for your time. And a lot of the credit of the research I showed you today goes to many of the people in our lab who work very hard to help advance our understanding of the prevalence, risks, and mechanisms for SUDEP.